Hello, and thanks for coming to our poster today. To start, I just wanted to go into a short background and motivation for our study, and then after I'll go into each panel individually. So as many of you are aware, stress granules are dynamic structures that form in response to a variety of stressors. Some studies have hypothesized that stress granules evolve into the pathological inclusions that are observed in ALS patient neurons and glia. However, the proteome of stress granules has not been completely defined. Recent studies have begun to study the proteome of stress granules by interrogating the interactome of G3BP1, which is a well-characterized stress granule markers. We've recently shown that Caprin-1 stress granules actually form independent of G3BP1, further motivating us to study the Caprin-1 interactome to gain further insight into the stress granule proteome. This figure is an overview of our IPMS experiments where A through D show the overall mass spec results and a representative Western blot confirming specific IP of Caprin-1. E is a Venn diagram comparing significant interactors of Caprin-1 in both the stressed and the unstressed condition, with a large overlap between significant interactors in both conditions. To get a sense of the function of the Caprin-1 interactors, we performed pathway analysis on both the interactors in the unstressed and stressed condition and found that these interactors annotated to the same pathways, specifically ribosomes, RNA transport, and the spliceosome pathway as shown in figures F and G. So therefore, we focused our attention on proteins within these pathways for downstream validation of our mass spec results. We also compared our stress list to RNA binding protein databases, and those results are shown in H here. So collectively, from all the analysis in F, G, and H, we derived our short list of 12 Caprin-1 interactors that we validated by immunofluorescence staining. Out of the 12 interactors that we validated, 4 of them colocalized with Caprin-1 stress granules in HeLa cells, as shown in figures A through D here on the left. We also tested these 4 interactors in both undifferentiated and differentiated SHSY5Y neuronal-like cells, and only SNRP200 colocalized with Caprin-1, as shown in figures E and F here. We also wanted to note that we also tested heat shock and osmotic stressors, and SNRP200 also colocalized with Caprin-1 under these conditions in all the cells that we tested. Based on these results, SNRP200 became our top candidate, and so we wanted to see if it was a component of the cytoplasmic inclusions that we typically see in ALS patient postmortem tissues. And indeed, that is the case here. We performed IHC on postmortem tissues from ALS cases, disease controls, and non-neurologic disease controls in both the spinal cord and the motor cortex. And as pointed out by those black arrowheads, we saw SNRP200 inclusions in ALS cases, but not in the controls. And at this point, I'll go ahead and stop here and thank you again for coming to our poster. Please feel free to reach out if you have any questions.